Hi, my name is Jasper, and for this project, I chose to research Bifidobacterium which is a bacteria in human breast milk. I observed kind of a few different ways to pronounce this word, and one of them was Bifidobacterium breve. So I'll be saying breve because it feels more natural for my mouth, but I've heard both. So I decided to create a brochure because I work as a birth and postpartum doula. So I wanted to create something that I could provide to my clients to give them like a little informational piece when it comes to breastfeeding, if my clients choose to do that. And so stick with me. I know it looks a little crazy because this brochure is flat, it's not folded up, but we will hop around here. So oftentimes what comes to mind when people think of choosing human milk for their infant is all the nutritional components that are beneficial to growth and development. But I feel like many things, we often kind of forget what's going on behind the scenes. And in this case, it's many bacteria, many different types of bacteria working to kind of create those nutritional interactions. A couple of the other bacteria that make up this microbiota are lactobacillus and streptococcus and many more. They all kind of make up what is known as HMM, which is the human milk microbiota. And Bifidobacterium breve is included in the HMM, and it's a probiotic bacteria that is commonly associated with the gut microbiome. That's kind of where it's most often observed and implemented into like foods and things to promote the microbiota gut health. So members of the genus Bifidobacterium are, like I said, one of the most common, found in the human gastrointestinal tract, but they're also found in other things like certain foods, waste products. So they're kind of grouped into multiple umbrellas, and Bifidobacterium breve is grouped especially under human residential Bifidobacterium, which I feel like is pretty, makes sense why it's categorized that way. When thinking, kind of looking more closely at this bacteria, like you see this little orange picture down here, we look at sort of the anatomy and more specific pieces. Bifidobacterium breve is rod-shaped, and it kind of creates these unique little shapes that are like V's or Y's. And it's anaerobic and gram-positive, and it's non-motile. And it interacts with other organisms by branching, which you can kind of see here. It does this little like branch and then into a Y. It mostly relies on complex carbohydrates for nutrition, and thus why it does so well in the gut. And it also has kind of an ideal temperature for growth, which is 37 degrees Celsius, which aligns perfectly with the human body temperature. So that's why, again, why it grows so well there. When we think more into the benefits, specifically in breast milk, there's increased resistance to infection, stimulation of the immune system, protection against cancers and different diseases, and bifidobacteria also secretes certain vitamins like B12. And then so on this like middle fold in the back, I have my resources, a reminder that while there's specific benefits to breast milk or human milk, chest milk, that fed is always best for infants. And I have a little blurb on the back of just the benefits of human milk, just kind of my knowledge from my job. Benefits the immune system, like I mentioned earlier, creates balanced nutrition, supports brain development, specifically the colostrum in the first few days, and supports the gut microbiome through beneficial bacteria like Bifidobacterium breve. Thanks for listening.
Hello, today I'll be talking about Bartonella Hensley, which causes cat scratch disease. B. Hensley is a small, rod-shaped gram-negative bacteria which favors aerobic environments. It belongs within the phylum Proteobacteria, and more specifically within the class Alpha Proteobacteria. B. Hensley is the most common among many species within the Bartonella genus. Anatomically, B. Hensley lacks flagella. Rather, it relies on the twitching of its fimbriae to propel movement. And here we can see a relative of B. Hensley, which is positively identified using the Warthin starry stain. Domesticated cats, particularly kittens, as well as stray cats, are the primary carrier. However, they are often asymptomatic. Humans contract B. Hensley through cat scratches and bites, and it is a fairly common disease with about 22,000 cases reported annually in children. Cats contract B. Hensley through biological arthropod vectors, specifically the cat flea, which spreads the bacteria through bites and feces. This is why strict flea control measures are so important among domesticated cats. Cat scratch disease often presents as mild to moderate flu-like symptoms, including fever and fatigue. There will often be a rash or papule at the site of inoculation, and swollen lymph nodes are a common symptom. Fortunately, for healthy individuals, the infection usually resolves itself naturally without the need for antimicrobial medication. Rarely, blood-borne infection may develop, which has downstream effects on the cardiovascular, nervous systems, as well as connective tissues. Interestingly, researchers have begun to explore the complex relationship between the brain and immune system through several small-scale studies, which have suggested links between B. Hensley infection and the onset of psychiatric symptoms, including psychosis. In one pediatric case study, treatment with antimicrobials resolved acute behavioral symptoms, which began rapidly following B. Hensley infection. It should be noted that this type of symptom is very rare. B. Hensley uses several strategies which contribute to its invasion and perseverance within the host, including the use of adhesion molecules, modification of the cytoskeleton of host cells, and survival within red blood cells to evade the host's immune defenses. B. Hensley also stimulates angiogenesis, or the production of new blood vessels. While study of these mechanisms could provide insight into future medical treatments, such as for coronary artery disease. For some individuals with compromised immune systems, rarely this can develop into unregulated vascular growth, which can cause serious damage to skin and internal organs. However, with prompt detection and treatment with antimicrobials such as doxycycline, prognosis is generally good. Thanks for listening.
Bacillus anthracis is a spore-forming bacterium that is best known for causing the disease anthrax. It is a gram-positive, rod-shaped bacterium that exists naturally in soil and can infect both humans and animals. What makes Bacillus anthracis particularly dangerous is its ability to form dormant spores, which can survive in harsh environments for years. When these spores enter the body, they can become active, multiplying rapidly and producing toxins that cause severe illness. Anthrax can be contracted in three primary ways: through the skin, cutaneous anthrax, by inhaling spores, inhalation anthrax, or by ingesting contaminated food, gastrointestinal anthrax. Though rare, inhalation anthrax can be fatal without prompt treatment. Despite its potential dangers, Bacillus anthracis is also studied for its use in vaccines and scientific research, due to its unique properties. Now that we know what Bacillus anthracis is and how it can cause anthrax, let's take a deeper look at how it affects the body and the different ways it can be contracted. There are three primary ways anthrax enters the body. The first is through the skin, causing what we call cutaneous anthrax. This happens when spores make their way into broken skin or a cut. At first, it might look like a small bump, but within a few days, it turns into a sore with a black center. While this might sound scary, it's the most treatable form of anthrax, and antibiotics can clear it up fairly easily if caught early. The second way is through inhaling spores, which leads to inhalation anthrax. When the spores are breathed in, they travel into the lungs and release toxins that can cause severe damage. This can lead to trouble breathing, fever, and in extreme cases, respiratory failure. Inhalation anthrax is much more dangerous than the skin form, and without treatment, it can be fatal. Now, let's talk about how we fight back against Bacillus anthracis. One of the most dangerous things about this bacteria is its ability to form tough spores that can survive in soil for years. To protect against it, we use vaccines, especially for those at high risk like military personnel and lab workers. The vaccine helps the body recognize Bacillus anthracis before it can cause harm. For those who do get infected, antibiotics like ciprofloxacin and doxycycline are very effective if caught early. In severe cases, like inhalation anthrax, additional treatments like antitoxins are used to neutralize the toxins. Thanks to ongoing research and public health efforts, we've made huge strides in preventing and treating anthrax, keeping it under control.
Now that I've shared some of the critical aspects of Bacillus anthracis, I'll take it from here and wrap up the final details. To start, Bacillus anthracis is a rod-shaped gram-positive bacterium that can form spores, making it highly resilient in harsh environmental conditions. It is classified within the Bacilliaceae family, and though its closest microbial relatives include species like Bacillus cereus and Bacillus subtilis, Bacillus anthracis is most infamous for causing anthrax in humans and animals. In terms of metabolism, Bacillus anthracis is aerobic, requiring oxygen for growth, though it can also tolerate anaerobic conditions. This adaptability allows it to thrive in various environments, contributing to its resilience. When it comes to microbial nutrition and growth, this bacterium primarily feeds on organic compounds, utilizing a variety of substrates for energy, which makes it quite versatile in different ecological niches. Bacillus anthracis also interacts with other microorganisms, forming spores that can outlast harsh conditions and potentially compete with or be acted upon by other microorganisms in the soil and environment. In terms of interactions with larger organisms, Bacillus anthracis has significant pathological impacts, particularly in humans and animals, where it can cause anthrax, a severe infection. Its ability to produce spores and toxins makes it a dangerous pathogen, capable of affecting both humans and livestock and contributing to its notoriety as a biological weapon. As for environmental impacts, these spores can remain viable for extended periods, potentially leading to outbreaks in certain areas under the right conditions. Overall, the role of Bacillus anthracis is complex, balancing between harmful pathogenic behavior and resilience that allows it to persist in nature. The bacterium has shaped both medical science and public health practices in profound ways, and studying it allows us to understand broader microbial dynamics. With that, I conclude this presentation. I hope you found this information informative. My name is Andrea, and thank you for watching.
Hi, everybody. Welcome to my presentation on chlamydia. We only have 5 minutes here, so I'm just going to get right started. Chlamydia trachomatis is the most commonly reported STI in the United States. And then Chlamydia trachomatis, the same genus and species, actually causes an eye disease called trachoma, which is going to be the leading cause of non-traumatic blindness in humans as a whole. So same organism causes two very, very relevant and significant diseases. Some characteristics of this group of organisms. Chlamydias are the smallest bacteria that are known to infect humans. They're about 0.2 micrometers in size, so very, very small. They are obligate intracellular parasites, meaning they have to live within the cells of their host, which are generally going to be mammal cells. And then they're also classified as gram-negative. Something interesting is that they don't contain peptidoglycan cell walls, despite the fact that they are classified as gram-negative. This is likely because they are obligate intracellular parasites. So because they live inside of host cells, they don't really need that extra kind of cell wall protective layer. And then they're also cocci, they are non-motile, and they replicate through binary fission. And I added a little bit on the side about the fact that chlamydia were actually once thought to be viruses. And this is because they're so small. They're much smaller than most bacteria. Again, they're obligate intracellular parasites, as most viruses are. And then they don't have their own system for generating ATP. So they actually, a lot of the times, are called energy parasites. So they do share a lot of characteristics with viruses, even though they are now understood to be bacteria. Something really interesting about this group of organisms, chlamydias actually have two forms. They have something called an elementary body and then a reticulate body. And the elementary body is going to be an infective and replicative form of chlamydia, whereas the reticulate body is just exactly the opposite. It's a replicative, non-infective form. And chlamydias can actually switch between these two forms. And so we're going to look at the image here on the right and kind of see how that happens. So In point number one, we've got the EB, the elementary body, which attaches to the receptor on the host cell. The host cell then brings the EB inside of a vesicle. And then now that the elementary body is inside a vesicle, it wants to replicate. And so then it turns into the RB, the replicative reticulate body form, copies itself a bunch of times through binary fission. Now you have the vesicle that's full of all of these cells, chlamydia cells. They can then turn back into EBs, and then they burst out of the cell and go infect other cells. So it's really, really interesting that they kind of switch between the infective but non-replicative stage and then the replicative but non-infective stage, and vice versa. They just keep kind of going through that. We're going to mostly focus here on Chlamydia trachomatis, a specific genus and species of chlamydia, because they cause two very significant diseases in humans that we had kind of talked about. So Chlamydia trachomatis causes the STD lymphogranuloma venereum, which is colloquially known as chlamydia, and then also causes trachoma, which is an eye disease. And basically what happens with trachoma is Chlamydia trachomatis is going to infect cells of the conjunctiva, kind of right there on the eye, causing an inflammatory response, can actually cause the eyelid to kind of fold inward on itself, and the eyelashes can actually abrade the cornea and cause blindness that way. Some more kind of interesting facts about Chlamydia trachomatis. So this organism only infects humans. Humans are the only host of Chlamydia trachomatis, and it's only going to infect cells that have a receptor for that elementary body. And these are primarily epithelial cells, so cells of the conjunctiva. They're going to be mucous membrane of the respiratory tract, and then also the urogenital area. And it's primarily going to spread through sexual contact, so through bodily fluids. Can also be through direct contact with infected tissue, either through a cut or an abrasion of some sort. Can actually spread also through flies and feces, and then during childbirth as well. And lastly, we're going to talk a little bit about treatment and prevention here. So treatments generally can involve antibiotics. So tetracycline, doxycycline, those types of broad spectrum antibiotics can also be treated with erythromycin and azithromycin. And for prevention, we have things like safe sex, kind of public sanitation, so making sure to keep feces away from living areas. Managing fly populations is also very important. And then medical care before childbirth I included because sometimes people are infected with Chlamydia trachomatis but are not aware and can actually pass that during childbirth. Coughing etiquette, just coughing into the elbow is important because Chlamydia trachomatis can actually spread through respiratory droplets. And then, of course, hand washing. That is all I have for today, but thank you so much for watching, and I hope you enjoyed and learned something new.
Let's go for a hike. Hiking is so beautiful, but there is a lot to watch out for that can be dangerous. Make sure to watch out for bears, snakes, and poison ivy. They forgot about watching out for me, Rocky the tick. I have Rickettsia rickettsii bacterium in my midgut and salivary glands, and I am ready to bite. This is my friend Rickettsia rickettsii. Hello, us are Rickettsia, are obligate gram-negative coccobacilli bacteria. Many of my Rickettsia family relatives use vectors like Rocky and other insects and rodents to transmit to humans and other animals to cause disease. This is because we are non-motile and cannot move without a vector or host. We can only grow and replicate in a host cell using the host's metabolic pathways. Here is my family tree of similar microorganisms to myself. We all have many similarities in common. For example, we all need to be transmitted to a host via a vector like ticks, rodents, or fleas. Most Rickettsia-like organisms are small rod-shaped bacteria. Our Rickettsia genus has 24 species classified into 3 groups based on their genomic, antigenic, morphologic, and ecologic patterns, which are the spotted fever group, SFG, the typhus group, and the Arbellii group. But we have our differences as well. Most of us cause diseases, though they are all different and have different effects on the host, while a few relatives do not cause disease at all, like Rickettsia canadensis and Arbellii. I use pathways of glutamate and glutamine utilization for metabolism to stimulate respiration. While I cannot use glucose, lactate, or sucrose to stimulate respiration, I also heavily rely on my host to provide me energy and metabolic pathways. I must use a host to access nutrients and be able to reproduce. When I am within my arthropod vector, like Rocky, I will interact and live within the environment of the vector. Since we are intracellular bacteria, we do not interact with a lot of other microorganisms other than the ones inside our host. Speaking of our hosts, they tend to be humans or some other animals like dogs, which allow us to attach, infect, and reproduce and cause disease. What great hosts they are. Now that we have introduced ourselves, let's find a host we can infect. Now that Rocky has found a host, we can enter and begin to replicate. Once we are inside the host, we can enter the circulatory system of the host. Our goal is to replicate in the host cell's cytosol, then to escape the endothelial cells to infect other cells. We are really not that bad. We do not secrete any toxins to harm our host, but our escape from the endothelial cells do cause the host damage. Rumor has it us R. rickettsii cause a disease the humans are calling Rocky Mountain spotted fever. But how silly is that name, as many hikers actually contact this disease from our vectors in the Appalachian Mountains, not the Rockies. The damage into the host's endothelial cells when us R. rickettsii as replicating include leakage of blood vessels into other tissues, which causes low blood pressure and lack of nutrients to the rest of the host's organs. This damage is the cause of the signs and symptoms associated with Rocky Mountain spotted fever. About a week after we enter a host, we cause them to have many signs and symptoms. These signs and symptoms include headache, chills, confusion, spotted rash that sometimes turns into subcutaneous hemorrhages called petechia, nausea, vomiting, and even respiratory and renal failure in some hosts. About 5% of cases result in death. Okay, so maybe we are a little bad.
Okay, so for my term project, I decided to do it on the bacteria Neisseria gonorrhoeae, and this is the bacteria that causes the STI gonorrhea in humans. This is the second most common STI in the United States, and there's an average of 82 million cases worldwide per year. So this is a gram-negative bacteria. Its shape is cocci that are in pairs, like little balls, as you can see in the picture. It has fimbriae that extrude from its cell membrane, and these act as adhesion factors, which allow these gonococci to attach to cells, typically lining the cervix or the urethra. And they're from the genus Neisseria, and this is the only genus of gram-negative bacteria that cause diseases in humans. And the other pathogen from this genus is Neisseria meningitis, which causes a form of meningitis. In terms of metabolism, these are obligate aerobes, and though they're generally considered obligate, they can use anaerobic respiration when needed by using nitrate as a terminal electron acceptor. And they are both catalase and oxidase positive. And they can ferment glucose, but no other sugars. And they don't really interact with many other microorganisms since they are a pathogen, and a normal and healthy human microbiome would not contain these. For my art integration project, I decided to crochet a little gonococci. So you can see them attached together in the middle, and then they have their fimbriae extruding from there. And I just used yarn, so they're a little flimsy, but that's the idea. And then it's pink because it's gram-negative. All right, so this bacteria is able to thrive within the body due to a few virulence factors. First, its fimbriae and some other cell membrane components allow them to adhere to the epithelial cells of our mucosal membranes. And then it also contains the enzyme protease, and this enzyme actually cleaves an antibody that acts as an innate line of defense in our mucosal membranes against pathogens. They also get picked up by neutrophils, and they are able to reproduce and survive within the neutrophil. And they can also be transported intracellularly by phagocytes, and that allows them to invade deeper connective tissue. In terms of symptoms, it varies a lot by person and by sex. In females, it's very common to be asymptomatic. It's about 50% of cases. And then when you do have symptoms, it can often be confused for as a UTI or a yeast infection. Males are a lot more likely to have symptoms such as painful urination or discharge, and this typically happens about 2 to 5 days after infection, but it can incubate for up to 30 days. So the current treatment is cephalosporins plus azithromycin or doxycycline. And a concern with gonorrhea in this bacteria is that it's developing major antibiotic resistance, and we're currently on our last effective antibiotic for this bacteria, which is clearly a very big public health concern because if it becomes resistant to this last group, then we won't have an effective treatment. And there's also no long-term immunity with this bacteria, meaning that if you get infected once, it's not going to prevent you from being infected again. In terms of prevention, abstinence, just refraining from sexual encounters. But if you aren't going to do that, just making sure to use condoms properly. And then I also think a really important part of it is just regular STI testing and education, having comprehensive sexual education plans in schools, and just spreading awareness on all STIs so everyone has accurate information. So that is it for my term project. Here are my works cited, and that is it. Thank you.
Hi everybody, this is Felicia, and my term project is on Salmonella, and this is an image of Salmonella enteritis, and I attempted to do artwork replicating, should I say, this particular image. This is my interpretation of Salmonella enteritis. Here it is. Don't judge me too bad because I had fun doing this project, creating this. So here it is. So a little key points for Salmonella is that it's gram-negative, motile bacilli resident bacteria that lives in intestines in almost all mammals, birds, and reptiles, and it's eliminated in the feces. It's the leading cause of zoonotic foodborne disease worldwide. Most infections are from food that has been contaminated with animal feces, but also contaminated water is a problem as well. And believe it or not, many have been contaminated with salmonella from their own pets, namely lizards, but also other pets as well. There are 2,500 serotypes of salmonella, and there is a strain specific for human to human, and it's called Salmonella typhi, and it is linked to typhoid. And there is a vaccine for it. When you travel to certain countries, it's required because they have a epidemic of typhoid fever there, and that is linked to salmonella. So you have to get that vaccine to help with that when you go to certain countries. Um, Salmonella causes fever, nausea, vomiting, diarrhea. What it does is it affects the cells in the intestines to where it can't absorb water. So you get dehydrated, and also it creates diarrhea. So also that is creating dehydration. And then there is a case in Dublin where the salmonella actually crossed the gut barrier and entered into the bloodstream. There is also cases where if you didn't get treatment soon enough, that you could lose your life because of these symptoms from salmonella poisoning. Salmonella is mesophilic, so it grows best in moderate temperature, not too hot, not too cold, and it creates a great hardship on the economy, on people being sick. You have to pay for medication. You're losing time at work. You're losing food products. Usually a great deal at one time, and that is hard on the consumer, and that's hard on the seller as well. It's like a big circle of just loss. And I also wrote a poem about it, and it's welcome to our public safety concern. Let's see what I can tell you about salmonella that you might want to learn. One thing's for sure, this will make you think. Be careful where you go, be careful what you do, be careful what you eat, be careful what you drink. Because if you don't, salmonella is waiting to give you some gifts of despair, like stomach pain, fever, and runs from you know where. Salmonella is a resident bacteria lurking in the gut, looking to be tagged in so we can act up. Time to take notice, time to be alarmed. How our food is being processed is causing us harm. Failing to do hand care when cooking our food is another reason we don't feel good. This right here is a worldwide threat affecting countries like Canada, US, Nova Scotia, and Quebec. Of course, you know there's more. Japan, Brazil, Australia, probably Ecuador. How can we get this under control? Let's see. Wash your hands, please. Cook your food well. Store it at a safe temperature. This is what I've been told that can get this under control. I hope you've enjoyed this short time together. Me, you, and salmonella.